Yes, sir. Sir, can we start the workshop?
Sure.
Yes, sir.
Now, am I audible? And am I? Can you see my screen?
Yes, sir. Sir, you are audible and your screen is also visible, sir.
Okay. Well, good afternoon, everyone, or good evening, as it will be where you are. I was told earlier on that there are some people from UPSIFs already in the audience, so I hope that I'm not going over things that I spoke to you before about, but I'm sure there are 149 people in the room at the moment, so I'm sure there'll be something for everyone. So this year marks my 40th year in forensic science. I started in Kent back in 1986. I had a short period of that in the north of England, in the Yorkshire area where I was born. So I suppose I've just been lucky to have been around forensic science at a very, very exciting time. The evolution of biometric databases with fingerprints, and in 1995, our database going live. So it's lovely to be with you all. I really do enjoy my time in India. I'm in India regularly and always love being with you all. So so this is my agenda for today. I'd like to try to tell you where it all began, and I can't see your young faces. I'm guessing they're young, but it started to be around 40 years ago, and talk you through some of my history, my background, tell you a little bit about where the techniques have evolved, where they've come from and so forth, and any changes in sensitivity and discrimination, and perhaps touch upon a few little bits of the future, particularly in respect of machine learning, large language models, and what those types of technologies are going to actually bring to the to the party. So that's what I'd like to get through today with you. But let's start really at the beginning. And I think we've got a little bit over an hour. I think I'm right in saying that we have about an hour to do this. So I'm going to hopefully try to keep to that hour. But to take you right back to 1987. And like I say, I can't see your faces. I'm guessing that there weren't many of you around in 1987. But that really, the key to telling you that story, the beginning of this story, is that it was the first time we actually identified anyone. Oh, my screen's just gone blank. Someone took control of the screen, and then it just went blank. Shall I start again? So I'll just go through where we've got to. So some of you might have heard of this particular person. His name is Colin Pitchfork, and he was the first murderer ever to be identified with DNA. And it began in 1987. Two young girls walking home from walking back home from school or from parties, that type of thing. And the first one in 1983, and the second one in 1986, so about 3 years apart. Now, bearing in mind, all we had in those days really was the standard serology tests, the ABO test, the normal blood grouping tests, combined with PGM, which would give us a figure, and that figure was, you can see there, it's 10% of the male population. There were semen deposited on both victims. Both victims had been strangled and murdered, and that's really all we had in those days. And of course, those techniques were good for exclusion. They were good to exclude a person from inquiries, but they were actually no good for inclusion. I don't know what the male population of India is, but you can imagine if it's 10% of what the male population is, it's not very good. It's not giving as many clues, is it? So as was looking at point number 4 now, as was common in those days, and perhaps still is to this day, these cases would run generally for about a year before the investigative leads started to dry up. So you would have an incident room manned by police officers and specialists, and after a while the calls into the incident room and the clues that were coming in would dry up, so that the hunt was called off. You can see there in 1984. So that was that first case. Then of course the second case was in 1986, and similarly another inquiry was started. And the key part of telling this story is that over the years DNA has had a little bit of a mixed reception, really. Some people think it's fabulous. I think it's fabulous. Other people think it's far too intrusive, and there's ethical problems and all that type of thing with it. But look at point number 6. Richard Buckland was a young man who actually confessed to one of the murders. He actually, I don't know why he confessed. Why do people confess? Is it because they're coerced into a confession? I really don't know. But he confessed back in '86, and it was Alec Jeffreys, Professor Sir Alec Jeffreys at the time, who was doing some work not too far away actually, where Narborough is in the county of Leicestershire. Alec Jeffreys was Professor of Genetics at the University of Leicester, and had actually been doing some experimentation with RFLP profiling, so some of the early techniques of DNA profiling. And so he was approached by the police service, and he said, okay, we'll give it a go. It's never been done before. It's been done in the laboratory, of course, but they decided that they would have a sort of, it's called like a dragnet inquiry, so where all the young men in a particular area would be gathered together, and would have a sample taken from them, a blood sample, and that was the very first time that type of technique was actually used. So all the people all the young men in a particular area of Delhi or wherever would be gathered together and would be asked for a sample. And so that's what happened. And in fact, just a slight aside, if there are any of you who have or want to buy the book, there's a book called The Blooding. It's quite easily bought. It's quite cheap actually nowadays on Amazon. It's called The Blooding. It's by Joseph Wembley. And the reason it's called The Blooding, it tells the story of this first inquiry. It tells the story of actually how this was done for the first time. As I say, it's by Joseph Wembley. And it tells you that whole story of actually how this was done in more detail than I'll tell you. So point number 8, Pitchfork managed to really thinking, well, I've deposited my biological material on both of these victims. Buckland, of course, had been exonerated. Alec Jeffreys had said, well, actually, the two cases are connected. But Buckland would only ever confess to one offence. He wouldn't confess to both offences. So, and it was Jeffreys who actually said, well, it's the same person who's committed both offences. So that very first case, called The Blooding, also had an exoneration. So the point I want to make with you rather clumsily, I suppose, is that DNA technology has the power to exonerate as well as inculpate, as well as identify people. So he knew that he was going to get caught. He managed to convince one of his friends to take the blood sample, or go along to the police station, like a little village hall, that type of thing, and take the sample for him. And he spanning some story that he was frightened of needles, frightened of injections, and so still, the case went cold for a little while because there's no match on the database. He knew that if he put his blood into the system, he would match. So he arranged for this guy to stand in for him, and it was only when a police officer's wife, overheard the friend of Buckland boasting in a pub one night how he'd taken the test for a pitchfork, She went home, told her husband, who was the cop, and then he was arrested. You actually see that the sort of latter parts of coming up to date a little bit with, he was released in 2021. So from 83, when the case happened, to 21, he was in prison, or shortly thereafter, sentenced to prison in 1988. Released on licence in 2021, and then recalled to prison for some offences. If you breach your conditions of your licence after you're released, then you're recalled to prison. And the key part of that story is that we found on the cold case programme that I ran many years ago that these people who commit these types of violent sexual crimes do not stop. They don't stop until you guys, you being the next generation of forensic specialists, you will stop them. That's what he looks like now, and of course life's not good to any of us, is it? So that's Colin Pitchfork as he looks now. So let's just take a moment to look at a summary. Coming right the way back up to date, or mostly up to date. What can we do now? Back in those days, the sampling that we had took in the region of months. It was run on an X-ray plate, run with agarose gel. I'm pretty sure that you probably all run agarose gels. It was called RFLP technology, restriction, fragmentation, length, polymorphisms, and it was really no good for building a database because it meant that you had to have a one-to-one comparison. You had to compare the crime sample with the suspect. Well, it was actually good for, it was very sensitive, and it was it was that very first technique. But it took about 3 or 4 months to actually get these profiles through. Fast forward now to look, some of you might have heard of rapid hit DNA technology, the technology that's actually able now to give you a profile from a reference sample, primarily from a reference buccal scrape sample, in the space of about 80 minutes. So if someone is brought into a police station and they say, my name's Bob Green, and they're not, and the sample can be taken, and it can be then checked against the database or against a family member, that can be done very, very quickly. So that's, speed has been, we've come to massively speeded up the process. And in fact, the standard processing time in the UK nowadays, just the standard time, if you submit a sample to a lab one day, the standard time was about 5 days. Some of you will have heard, because I know when I come to India, you always ask me about next generation sequencing, whole genome sequencing, that type of thing. And next generation sequencing is the new kid on the block. I'm going to speak to you more about that later on. But there are a number of variants of next generation sequencing. It's actually not next generation at all. It has been around for a good few years, but we've just started to use it in forensics. But it's that type of technique, next gen sequencing, that's used with, very sadly, the war in Ukraine is obviously killing many, many young men who need to be identified. They're often buried in very bad conditions, and next generation sequencing enables us to deal with, with material we could never ever deal with before. We then got to improved mixture analysis, and this really is where I think machine learning will continue to play a real part. We can actually identify DNA now at far lower levels than we ever could previously. Back in the day, when we first started, you would need a piece of DNA round about this size. I don't know if you can see my fingers, but round about an inch across, or perhaps a little bit slightly less. But that's the amount of DNA that you would need. The threshold of detection now, the standards, the standard at which we will actually start this whole process is 500 picograms. Many of you will know that a picogram is a trillionth of a gram, and we need 500 of those to actually get the whole process going. So 500 picograms, and on occasions when we have a nice clean sample, we can actually go down to 200 picograms. So what that means is that any sample, anything that really touches your skin, any spectacles, any watch straps, any glasses, that sort of thing, anything that touches your neck, you'll actually be able to recover a DNA profile from. Our ability to take DNA has massively improved in terms of sensitivity. However, our ability to interpret what we actually have there at the end of it is perhaps sometimes lagging a little bit. We just pick up lots and lots of mixtures. Some of you will actually know this if you've done any DNA profiling. So, improved mixture analysis. I'm told that the audio is not working properly. Let me see if can I turn it up? Is that working properly now?
Yes, sir, properly. Sir, you're audible.
Okay.
Working properly.
Okay, thank you. So, improved mixture analysis is where we will be going, I think, more with, we have techniques now called probabilistic genotyping software, and I think that will become more and more, come more to the fore. I'm going to explain to you that in a little bit later on as we move through the presentation. But we've massively increased the sensitivity. I can still recall my very first DNA case back in around about 1988-89. It was a young girl who'd been raped, and the offender had actually left her dead. He thought he'd killed her, and we actually found the condom. And back in the day, the pitchfork case had just happened, and we thought this really is going to be the breakthrough. So we have a condom full of biological material. Sadly, it didn't give us any link to the offender, because literally we didn't have enough to start with. Fast forward now, 500 or 200 picograms actually starts the whole process going. So we have some new techniques in terms of genetics and genealogy, which I'm going to come on to tell you about. Forensic investigative genetic genealogy, you may have heard of. FIG, it's sometimes referred to as. And I've got some case studies as we go through. So the other thing to mention, guys, is that there are some hyperlinks in this presentation. I'm pretty sure I've got some hyperlinks in this presentation. So if you have your phone handy, I can go through some of those with you. I can actually give you some of the materials, but if not, let me just make sure I've got the right presentation with me. Okay, guys, so let me just move on. So nowadays, the strengths are high accuracy. It's sometimes referred to as the gold standard of forensic science. I don't think it is. I think it's a really great standard. But of course, fingerprints can still do lots and lots of things that often quicker, often lots cheaper. It can give us an individual identification from where we actually never had this ability to do that before. And hopefully, as another case studies I'm going to share with you, some of these stories will be embedded with those case studies. We can look back at cold cases, some cases that happened many years ago. At the program I actually ran in the Home Office when I worked in the civil service. Look at these cases and realising, amongst other things, that these people do not stop. They come due to offend into their late adulthood. And the final bit, of course, is the exoneration aspect of these cases. So where are we nowadays? Well, thanks for the emojis, guys. But the added sensitivity is now giving us problems in terms of contamination and in terms of mixtures. So contaminated samples, mixtures are of low quality, because remember, beforehand we wouldn't even have started with samples of this level. We wouldn't have actually started. They would have just said, that there's not enough to begin with, or in fact it's too degraded. Where now we're actually recovering these small amounts of bone from Ukraine, analysing the bones actually done in the Netherlands, run through next generation sequencing, and finding how to try to repatriate the bones. So we've got contamination risks, and for those of you who are new to the profession, please guys remember that contamination and anti-contamination procedures now are taken to a completely different level. If you don't wear personal protective equipment, if you don't wear gloves, masks, full coveralls, if you sneeze or poke over an item, you will actually contaminate it. Remember, there's enough in spit in your breath to deposit 500 or 200 picograms often. So we get lots of complex mixtures, which is really where AI and that generation of technology is going to come in. And of course, we've got ethical concerns in terms of privacy. And just hold those thoughts, please, for a minute, because I'm going to tell you some of these stories as we move through, and hopefully all of this will come into fit into place. I love the emojis, guys. You're making my head swell. So where do we get the DNA that we deal with, the DNA that we actually use on our database? It's nucleic DNA. It's from the cell, so it's all cellular material. Sometimes we recover DNA at such low levels that we actually can't. The word we actually use is attribute. We can't attribute this to one particular body type, body fluid or another. Looking back at my early day with the condom, in the semen in the condom, and the amount of material we had to look. Of course, we have to look for the nucleus. So we look at nucleic DNA, but there are techniques that we were experimenting with as we went along that looked at the female component of DNA, and you've all probably heard of that. Mitochondrial DNA is the female component, a little bit like the YSTR is for the male component. The female component we actually get from the YSTR from the male. So of course, one of the first things we need to do is to find the nucleus inside the cell, cytoplasm, the mitochondria, and everything, and we have to break open the nucleus. We call that cell lysis, and then we find two pairs of chromosomes at various locations on those chromosomes, areas that we refer to as polymorphic. It means that there's many changes, lots of variation. We call them polymorphisms, and we find let's call them markers for the sake of this session. There are DNA repeating sequences that really don't code for any. We call them autosomal DNA, and they give us. And that's where we find them, from the human to the cell, to the nucleus, to the chromosome, and then of course to the double helix, which you'll see there. And all of our cells have DNA, except red blood cells, which really, guys, is why perhaps semen, that type of body fluid, is a much richer sample. Often, speaking nowadays, there's enough DNA in blood samples to get something from it, but we can't get DNA from red blood cells, and it makes us really who we are. There is some research being undertaken, I see it pop up from time to time, where the scientists now are experimenting with phenotypes. So we're looking primarily, exclusively at the genotype. So your genotype, the genotype you inherit from your mum, from your dad, and you see we get half from mum, half from dad. And we want to hold that thought again, please, because there are some aspects of this we're going to touch upon with some real life cases. And of course, then you have similarities between siblings. So some of your base pairs you will actually share with your siblings. So nowadays, in DNA profiling in the UK, we have a technique called DNA 17. It means there's 17 strands of DNA, 17 from mum, 17 from dad, 34 in total, and it's quite possible that that I would just happen to share some of those with my sister, or you might share some of those with your brothers and sisters. You will be a complete carbon copy, half and half of mum and half of dad. So 50% of your profile, your base pairs will come from mum, and 50% from dad. So these are the types of crime scene sample where you would find DNA. There used to be envelopes, there used to be stamps. Our stamps, you used to have to link them in the past. Sadly, those days have gone now. But we actually can get it we find DNA in many areas. And we can actually find it now in tinier and tinier amounts. So when I said to you we can't attribute this, the condom, we could actually attribute that to, we've got semen in that condom, we've got a blood sample. But sometimes if I just touch this laptop screen, I'm actually going to leave my deposit on the screen. In fact, even as I'm speaking to the camera, I'm depositing DNA on the screen and the keyboard. So the new kid on the block now is what we refer to as cellular material. We can't really say what it is. We recover it at such low levels that we really have to just recall it cellular material. We can't say it's blood, we can't say it's semen or saliva. We have to refer to it as cellular material. But we can actually get it from all of these substrates. So guys, one of the things I do with my students, and this is really taken from some of my teaching slides, and actually get my students to actually look back on some of the things we've said. I know from teaching lots and lots of Indian students over the years just how good you are at these these types of thing, but can you go through perhaps these questions, because sometimes you'll be sitting in a briefing, you might be being called to a serious crime, the senior police officer might be asking questions, he might have told you about the case, and you then have to, he might ask you a question, and then you're better prepared really to listen, to pay attention. The key to our profession is having that attention to detail, that precision, and someone says it's A, it was A. So anyone want to have a go at number 2? What was the outcome of the confession made by Richard Buckland? Was he convicted? Was his conviction found to be false? Buckland was found to be false. He was the guy who Alec Jeffries said, well, the two cases, Bourne, Ashworth and Lindemann, we have the same male biological material on both of those victims, but it's actually not, it's not Richard Buckland. When was Colin Pitchfork arrested in connection with the crimes? Does anyone remember? Who's got number 3? I know you guys are great at this. You're much better than my students are. So that third one it's C. It's September the 19th, 1987, which of course is many years before you were born. So this is the type of thing that I do in my lectures back at Kent. Now this is, I'm hoping some people have actually put that the audio is not very clear. Some people have actually put that the slides aren't very clear. So I'm hoping that you can actually see these slides well at your end. They appear wonderfully clear at my end, but of course that's to be expected, isn't it? Thank you, Simi. So this is the evolution of DNA, the technology of DNA from 1985 onwards, from the early Pitchfork era. And some of you, if you Google Alec Jeffries, Professor Sir Alec Jeffries, if you Google his name, you actually see a rather an iconic picture of him holding up an autoradiograph, the type of thing that you would have if you break your leg, God forbid, and it's a sort of an X-ray picture. So those are those early days, and those early profiles were like barcodes. You sometimes see them referred to as barcodes. Then in 1990, we had, so that first one was multi-locus probing. Multi-locus profiles are chemistries. I'm just thinking of the proper word to use now. things will actually bind to to multiple loci in 1985. In 1990, we had markers that would just bind to one single locus. So that was the 1990s, and we were starting then to think, actually, if we can move to the next generation of multiplexing, getting us away from the So the first thoughts back in 1994 were to have a technique called quad. By the very nature of its name, it meant that we actually targeted 4 sites. There's 4 sites, 4 from mum, of course, 4 from dad. So it really didn't give us much discrimination power. It was actually never actually taken into operation. What actually did start the database going was STR DNA databasing using SGM, second generation multiplexing. Second generation because it came after the multi-locus probes of the earlier years, and second generation multiplexing had 6 sites. 6 sites equals 10 markers. Remember, we get 6 from mum, 6 from dad. So it's like having a lottery machine, a lottery tumbler that has 12 numbers in it. Can you see that it's much more discriminating? You actually can't accidentally pull the right numbers out. So that's what we went live with back in 1995. Great fanfare early in 1995. The database went live. We put together the new legislation, and everything seemed to be going okay until 1999 came around. And what we were starting to find as adventitious matches. There were matches that were just happening at random. Let me just go back to the analogy with the lottery machine, please. If I have a lottery machine that only has 4 balls in the pot, there's a good chance that I can bring the right numbers out more easily, isn't they? Are you with me? I'm hoping you are. If you have something that has 16 markers, or on this occasion it had 10 markers. 10 markers equals, of course, 20 sites. It doesn't. It's a lot easier to get a false positive. So that was 1999. Then in 2000, some of you will, well, I'm guessing many of you will know of the technique PCR, polymerase chain reaction, very simply referred to as the genetic photocopier. I'll explain it to you in a moment. But that, in those early days, would run for 28 cycles. 28 cycles of heating and cooling, denaturation and extension and so forth. I'm pretty sure that this terminology you'll appreciate, you'll know. So in 2000 we said, well, actually what will happen if we go from 28 to 32 cycles? Remember that the PCR will actually grow up the DNA sample exponentially. I'll explain that to you in a moment. So we went from 28 to 32 cycles, and we could get far more out of less and less material. So that was the evolution of from the 2000s. In 2002, we had a database built, and we said, well, what would happen if we have a near miss? What happens if we have a near match on the database? It's not a match. It bears some striking resemblance to a family member, to one of your family members. Is there anything we can actually do about that? And once again, please hold these thoughts, because I'm going to give you some case studies of actually how we use these for the very first time. And then, so familial DNA was in 2002, and then that was all the way through really to from 2012 to 2021, when we realised once again that we had to put more lottery balls into the machine, because we were starting to actually get potentially some false matches out of the database. This is how STR, short tandem repeat STR profiling, works. STR profiling is the mainstay, the backbone really of forensic DNA. we do have some other techniques. Some people might have heard of single nucleotide polymorphisms, SNPs. But for the backbone of this type of technique, we actually use short tandem repeats. Of course, we have to extract DNA to start with. We have to extract the DNA, separate it from the substrate. Some of you actually may have done even some extractions at college or university. It's essentially a case of washing out the DNA with a detergent. Some people are putting it in the chat. You want a feedback form. I don't have a feedback form unless the organisers do. I don't have a feedback form, but if you want to send me a message in the chat with your email address, I'll gladly follow up on anything that you want to follow up with. So we first of all have to extract, then we have to quantitate. This is the test that was actually done at the time. It was a separate quantitation, and it's at this stage we actually determine have we got 500 picograms. The previous technique was at 1 nanogram. So at that stage, have we extracted 1 nanogram or 500 picograms enough to actually take us to the next step of the process? So quantitation nowadays, some of you will recall that we can have a technique nowadays called real-time quantitation, real-time PCR, which is actually combining the extraction and quantitation procedures together. Okay, and then of course we've got the amplification process, which I'm going to go on to next, PCR. And then of course you have the problem of separating these DNA fragments by size. Again, I don't know if coin sorters are commonplace in India, but I was only in a supermarket yesterday where you can actually throw your loose change into a machine in the supermarket, and it will tell you how much you've got there. We'll separate them all out into various denominations, so 10 rupee, 50 rupee coins, that type of thing, and it separates them out by molecular weight. So the small bits will fly through the gel. So that black picture on the middle of the screen there is an agarose gel, and we're actually applying some electrical current to the top of that gel, and we actually put a positive electrode to the top, and then it draws through the DNA fragments by size, so that the smaller bits fly through more quickly, and actually give you that separation. So it's separating out this by size. Nowadays we do this with capillary electrophoresis, but that final step of the process is in the interpretation piece. Someone's actually put in the chat when the slides are not displayed properly. Some are saying that when is the slides coming to an end? I think I've got a little bit more to go yet. I'm sorry you can't see the slides very clearly at your end, but certainly they're clear at my end. So this is PCR. It's a piece of equipment that heats up, denatures DNA. DNA is not thermostable, some of you will know that. So if you heat DNA up, it will denature. It falls apart. The bases will come away in solution. The nitrogenous bases, the structure will all fall apart in solution. And if we do that one cycle, can you see that we get, we've got one copy, we do it twice, we get 2 copies, do it 3 times, we get 4 copies. So that's what I was mentioning when I said we ran it for 28 cycles. 28 cycles it used to be. The current technology is run through at around about 35 cycles, I believe. So can you see that we're starting with much smaller amounts of DNA, and we're actually growing up the component parts of that. So what did we find as we went through this? That's a little bit the end of the science bit. Well, when we built our database, we found that we were getting matches of 3 types. One is the scene-to-scene match. I've used actually 2 houses here to depict burglary scenes, so we actually got the same DNA at both scenes. But of course it was the same type of scenario with Dawn Ashworth and Linda Mann. We actually found that same biological material at on both victims. So we can find a scene to scene where we actually don't know who the person is, but we actually got these two cases linked together. We can have a person to scene, pitchfork, and those two murders, and we can have a person to person, where the person is perhaps given a wrong, fictitious name, and as given a fictitious name, and it actually turns out that he's not the person who he said he was. That very first case with Pitchfork, where he arranged for someone to stand in for him. So we found, by running our database, by having our database a few years, that this is what we found about the individuals. Again, I'm guessing that this is the same in India. Every time I come to India, I read the English print papers, and I realise that you have quite a problem sometimes with sexual offences. We have found the same, that we have lots of sexual offenders. Rapists have 90% previous convictions for a lesser offence, so these people will actually start with a lesser offence and then build up to this crescendo. Armed robbery is greater than 50% of a criminal record, and serial offenders. So once again, your product as forensic scientists is not measured in single numbers. It's measured in many different, many magnitude of different offenders, because you will actually capture these serial offenders also. We found also, this was a study that was done by the National Improvement Agency, and they said that DNA was being used in greater than 95% of all murder cases. Fingerprints used in just under 3/4, and interestingly, just over a quarter of murders we used where footwear was used. So it's a lot more. You make it more of a contribution than perhaps you thought. So this really is what it means, or it meant back in the day. That these figures now have probably moved on a little bit. But if you have 275,000 burglaries, which we did have at one time in the UK, I don't know what it's like in India. So you could actually build a model that shows you how many matches you're likely to have. Let me just look at the chat. Someone just put something in the chat. Chewing gum was a new one for you. No, we get good results from chewing gum. We get good results from cigarette ends. Really, anything that really touches your skin. So how much DNA do we actually require to perform these tests? So all this nowadays will start with the standard threshold pico. That was halved from the previous technique. So remember, we had second generation multiplexing, that 10-plex marker, 10-plex, 20 markers. That on the latest iteration went up to 34 cycles, 34 markers. So DNA 17, we get 17 sites, giving us 34 possibles, both from mum and from dad. And each of those, we would actually need 500 picograms is what we call now our threshold of detection. If the sample is nice and clean, if it's a nice, good quality sample, we can actually go down to 200 picograms. 500 and 200 picograms, guys, you can't really see. It's the type of thing that, as I said, is on the arms of your spectacles, on the nose pieces of your glasses, on your watch strap, things like that. So we can. The problem now is not in terms of being able to recover DNA, but it's in terms of being able to interpret what it means. So we also found back in 2001 that it wasn't necessarily just picking up violent criminals, current violent criminals. This is a guy called John Wood. He was sampled for stealing a very tiny amount of goods from a shop. The samples had already been from the rape and indecent assault of two young girls in 1988, before the database went live. Those samples were loaded to the database and were stored. And when the database came around in 95, Wood matched immediately, and he was sentenced to 15 years. So it rather smashed the myth that that we that offenders have this specialism in offending. They don't. They'll have a go at most things. Then we really touched upon those very early days of mitochondrial DNA, where we were looking for not just the nucleic DNA, but is there anything we can actually say from the mitochondria? You probably all know that the mitochondria is the energy cell of the energy part of the cell. You can see that the nucleus is in the middle there, where we actually get these short tandem repeats. But there are lots and lots and lots of mitochondria, and we started to experiment with mitochondrial DNA, and particularly this was one of the very first cases that was done during my time at the Forensic Science Service. Some of you may have heard of this family. You put me a thumbs up if you've heard of the Romanov family. They were the Russian royal family, and they were executed back during the First World War, back toward the end of that First World War from 1914 to 1918. They were assassinated by the Bolshevik, who'd wanted the end of the royal family. And for many, many years, these bodies, they were executed in Katerinburg. All of the family were executed and were thrown down into pits, and then I think they were eventually found in a deep well, but were left there to rot forever. And so over the years, people have come forward to say, we're relatives of the Romanov family, because the Romanovs were a very wealthy family, and who were the Romanovs. So the Russian authorities approached the Forensic Science Service and said, could you help us with identifying these very badly degraded bones? Can you tell us, were they or were they not the Russian royal family? And so, in a similar way to why STR profiling nowadays, where we actually look at the male component, this was how the family tree was built with the Russian royal family. So toward the left-hand side of your screen, you've got Nicholas, and you've got the Tsarina Alexandria. You have the children, Alexei, Maria, Tatiana, Olga, and Anastasia. And all of those have the same mitochondrial DNA as the Tsarina Alexandria. Not the same as Nicholas, but they all share that same mitochondrial DNA. And we use mitochondrial DNA in the day because it was more resistant to degradation. But the key to telling you this is that we come up to the point of what we refer to nowadays as that point of common ancestry. You all know that you have two sides of your family tree. You have mum's side and you have dad's side. And we work up one side of the family tree and down the other side. So we then found that the same mitochondrial DNA, same maternal DNA, let's call it, with Princess Alice, who was the second daughter of Queen Victoria. And I've been over to India. I've seen some statues of Queen Victoria, so I know that she had some influence in India. Who then had the same mitochondrial DNA as the Princess Victoria of Hesse, who once again had the same as Alice of Battenberg, who then had the same mitochondrial DNA as Prince Philip, who was alive at the time, our late Queen's husband, because of course you have to have a living relative to actually get the DNA from. So he was the missing link, and a sample was actually taken from Prince Philip, and he had the same mitochondrial DNA as all of those victims in the well. So that was proved conclusively, and the Russian royal family are now buried in like a sacred vault in a cathedral in Russia. So that was the very first case we actually looked at for mitochondrial DNA. The second criminal case we looked at, which was not a criminal conviction in a sense, because it was this guy had actually, he died by the time people got round to him. He actually died in 1990. But he was referred to as the Saturday Night Strangler. He was a Welsh serial killer, you can see there, and he was responsible for the rape and murder of 3 teenage girls in 1973. We think it's widely recognised now that he did far more murders than, because they don't often stop with just one murder. But he targeted 16-year-old girls, luring them into vehicles. They were hitchhiking. They were filming a lift on the way back from going out for the night in the town. I can't see how many girls there are in the addressing the girls primarily, but I'm sure that your mums and dads have told you never to get into a vehicle with anyone. You can just give me a thumbs up if you've ever been told that. But anyway, these victims, they got into the car. They were murdered, and for many years we actually didn't know who this was. Remember, 1990, we don't have a DNA database. We don't really have much to go on at all. But this was the first time we actually used this to identify a person. Like I say, he died by the time that we got around to this, and we identified him through his son. His son was he bore the seeds of his father. He became a criminal. He committed vehicle crime, and afterwards we were able to actually look at his DNA profile and say, well, it's actually not the same as what was on those victims all those years ago, but it does bear a striking resemblance. So the victims who were sampled all those years ago were samples that were retained, and the son of Joseph Capon bore a striking resemblance. He would be half, wouldn't he? His profile would be actually half of Joseph Capon. So we would actually have some markers to start with. So he was born in 41. He died in 1990. He was a dreadful individual, very nasty, sadistic, violent man. And this was the very first time this familial searching and had been actually done on these types of case. So she was age 16. Sandra Newton was one of the victims. Remember, guys, that the other point to mention here to you all is that you are all working in forensic science. You're all doing almost like God's work, I would say. You're making life better for victims and their families. So she would now be an elderly woman, wouldn't she? She'd be a mum, a grandmother. So this is important. It's important to get this right. So this is how we've searched for family members. We can do two searches. One is for looking for the relationship between you and your mum, and your mum and your dad. So it's that sort of vertical relationship, the genetic relationship between parents and children. I'm guessing you can all give me a thumbs up if you've got brothers or sisters, and you will share that some of your profile on the x-axis, on the horizontal axis. So you will all have that type of that relationship. People are still asking for the feedback form. I'm sorry, guys, I don't have a feedback form, but I'm told the organizer will be able to give you a feedback form at the end. So what happened with Magnum was these cases remained unsolved for many years. The database went live in 1995. Then in 1998, samples were loaded to the database. Sadly, no match. We thought, blindly, we've now got this database, multi-million-pound investment in DNA technology, and we don't have a match. And we didn't have a match because we actually didn't have anyone to sample against. We didn't actually have someone to match against until it was decided that we'd actually look for some family members, and we looked for. We did this first familial search. To that first familial search, and we asked, are there any close relatives? We got 22,000 potential hits, and then we looked at the localities of these, where they were these people local or not. And as some of you might know, you can only build up so much of the profile. There will always be some bits of the jigsaw puzzle missing. Remember, Joseph Capon, the person, his father had died, so we actually didn't have his profile. We had Paul. It was Paul, I think his name was, the son. We had Mum, so we had some markers from Mum. We had some from Paul, and so we would have various bits of the jigsaw missing. So ultimately, we had to look to get the body exhumed. In the UK, we bury some of our bodies. It's becoming more and more common nowadays to cremate bodies. I think there's more of a preference for cremation in India, but over the years, lots of bodies were buried in graves. So the exhumation took place in 2002 on a very bleak, windy, raining night in Wales, and the bodies were, as I recall, buried in a stack of three. So the deepest body in the grave was Joseph Capon's father. Then there was Joseph Capon, who was the middle body, and then on top, one of his uncles had been buried. So that there were three bodies in this grave. And the true story, as Capon was being, as his coffin was being brought out of the grave, in that you can imagine, a pitch black, dark night in Wales, raining and thundering and lightning, and just as the casket came out of the ground, there was a massive big clap of thunder and a lightning strike, and I remember someone reporting that we said that that was evil being unearthed. So it must have been quite alarming for people. This is another case using this type of technology where this is a guy who was not on the database, on a DNA database, but he was a man who had been, in his younger life, had been committing a series of increasingly violent sexual crimes in the Yorkshire area, and we couldn't know who it was because, of course, he didn't match against anyone on the database. He was a very well-respected businessman, and actually he was traced through his sister. So we had an unsolved samples from all of these victims. We had a profile that looked similar to his. That was from his sister, who had been arrested for drink driving, and that's how James Lloyd was captured. We also found this, guys, and please take this forward with you throughout your careers if you can, to look at the offending patterns of these individuals. They will not stop until you stop them. That's the key message here. And if you look at that Gaussian curve, that bell-shaped curve in the middle, that's the standard pattern of a normal, shall we call them that, normal type of offender, who will start to offend in his early life, crescendo around about 21, 22 years of age, and then drop down the opposite side as they learn a little bit more sense, as they very often get into a relationship. So that's the normal pattern of offending. What we found with the programme of cold case work that I led in the Home Office was that the sexual and violent offenders actually continue on this increasing trajectory, this line. You can actually see that it says violent and sex offenders. So doing these things more quickly give you less chance, prevents lots more crime. So you can see on this next slide, if we do this more quickly, we take away the opportunity for these people to commit these crimes. If we don't do it so quickly, we get more crimes and more victims. So this was my the project that I led on behalf of the UK government. It was called Operation Advance. That's what we found when we actually looked at these samples. So these were people who had their samples taken in the early days of the DNA evolution, and actually the DNA was not compatible with the database as it stood at the time. So I was approached to say, look, can we look at these samples again under the new technology, and let's reanalyze them. Let's load them then to the database, and let's see what we get out of it. And we actually found lots and lots of people who otherwise would not have been captured because the samples had been taken. They'd been taken on those early years of DNA, but wasn't compatible with the database as it stood at the time. So there's lots and lots of learning we found out from this. 90% of offenders continue to offend after the rape that they were convicted for. So if we could have done this sooner, we would have solved lots more crime. The average number of convictions per offender was 10. The average number of offences per offender was 22. So these people who have this predilection for these types of crime will continue to offend until you guys, and I'll repeat it again, until you people stop them. Moving toward the end of these cases now is this case, Antonio Emilia. Some of you might have actually even been to London. If you've been to London and you've travelled in through Heathrow, you'll know that there's a big road, circular road, orbital road that actually runs around Heathrow, runs around London. And Antonio Emilia was a man who's a lorry driver, and he was committing his offences all the way around this M25 motorway. So this is when these offences were solved ultimately after all those years. As you see, the first offence was in November 2001. It was in Ashford, not too far away from where I live at the moment. But going out, let's say it was a lorry driver, so he was able to travel lots of distance. Can you see the ages of the victims? That first victim was aged 10, then to 26, then to 30, and 18. The last offender, once again, a 10-year-old, a 14-year-old girl, and 13 years of age. So that stresses really, once again, guys, how we all have to work together at this. Because what I'll say to you here is that first victim in November 2001, we could have never prevented that with the database, because we always need the first sample to match against, don't we? So that first crime is always, perhaps always doesn't go unsolved, because of course we capture it very quickly. But what it does mean is that we always fall short of one. But what we would have done if we'd have had that sample if we would have had Emilia's sample on the database at the time, we would have actually captured all of these people further down the track. And this is a case where Emilia's sample should have been loaded onto the database, and it wasn't. It was a mistake made by the police service, and it just hadn't been loaded, and so that gives you, that spells out that sort of trail of destruction that happened afterwards. So, God forbid, if you'd have been the mum or dad or the brother or sister of that person at 18, or that 52-year-old woman would have been your mum, can you imagine how you'd have felt when those types of mistakes happened? And of course, those are mistakes that don't impact on they're not to do with the science at all. The science works, but it's just the application of science. So let's come back up to speed now. Let's look where we are now, coming toward the end of the presentation. You'll be glad to hear. So from 2017 onwards, remember I talked to you a lot about next generation multiplexing. I said that there were new technologies on the horizon. Well, it wasn't necessarily just about number 3, next gen sequencing. It was about lots of other techniques. Enhanced capillary electrophoresis. So we'd gone from those early days of gels through to various iterations of capillary electrophoresis. By 2017, we've got this piece of equipment. We've got rapid hit DNA technology in 2017 or onwards. So these things it's really not new at all. And then the sequencer we need for next generation sequencing is at number 3. And so that's those are the improvements in technology. So But you can see now how things have become more and more sensitive. On the one hand, this is of course fabulous, but on the other hand, of course, it does cause some difficulties, as I've mentioned. We now get lots of incomplete profiles, lots of mixtures, and we do have some difficulties all the way through. Sensitivity and discrimination, I think we've mentioned already. Those early days, my condom days, those very early 87 days, 1987, 88, we would need a blood stain the size of a 2p coin. The starting material now is much less, which causes sometimes a problem because we can't attribute DNA. We can't attribute it to a blood sample, to a semen sample. We just have to say that we've swabbed a door handle, and we've developed a DNA profile, and it came from genetic material. Some of you will actually travel by train or by tram. I know trams are quite popular in India, and some of you will actually hold the handle as you on the tram. Imagine you've held the handle, you've deposited your DNA, or the person before you deposited his or her DNA, and then there's been a transfer. So the 500, 200 picograms level is that level of sensitivity. Along the way, we got caught out a little bit with low template DNA. This is a picture from a very sad bombing case during the years of the IRA bombing in Ireland. We were starting to experiment there with DNA, with low template DNA, using those enhanced cycles of PCR. That's not the only way in which you can actually do this, grow more from less and less, but it was the way that we did it at the time. And the low template, or low copy number, it was called in those days, took the standard test. You can actually see here we weren't growing very much at all, but repeating those sequence of heating and cooling, denaturation, extension, and so forth, we can get more and more out of less and less. And the point to make here, guys, is that the standard tests of today give you almost this level of replication. So not only are we picking up DNA at very much tinier amounts, but the replication process is much more sophisticated. So if you're not careful, you will contaminate. Just to give you an indication, you can actually see there that what this really refers to a single grain of sugar, but hopefully you can actually see that this is rather now a double-edged sword, so we can actually recover DNA at far greater levels than it's outstripped our ability to interpret, I suppose. We can't attribute, as we mentioned already, and we will now get mixed samples, so mixtures of samples where we can't determine who is in the mixture and who isn't. This is what I mean by sensitivity before and after. So low sensitivity in a fictitious crime scene, that's the amount of DNA that will be available. With high sensitivity, can you see the difference? And of course the implications. Please, guys, if you ever see anyone in your country examining a crime scene without full protective equipment, go up to them, tap them on the shoulder, and say, you're storing up problems for the future. So we mentioned DNA 17 is much more sensitive. It's much more discriminating. We've got more sites. We went from 10 to 34. From 20, 10 from mum, 10 from dad, to 34. That's 17 from mum, 17 from dad. They're more sensitive. They work better nowadays with degraded and inhibited samples. Yeah, inhibition really is an artifact really of PCR. So something that's inhibited. Back in the day, were things like denim jeans were a notorious inhibitor of DNA. Back in the day, cigars were a notorious inhibitor. So you could actually recover the DNA, but when it came around to doing the PCR process, you'd find that the sample was stifled. Degraded, of course, is where we have samples that are are just degraded by sunlight. And I know that in India it gets very, very hot. So those of you who are going to work out at a crime scene, make sure that if you do see a blood sample or a body fluid sample on the ground, recover it as quickly as you possibly can. Because of course, the DNA will be degraded by ultraviolet light, it will be degraded by the heat, it will be degraded by microbes in the soil that actually love to feed on DNA. So recover that as quickly as you possibly can. But the new multiplexers are giving you much, much more opportunities if you can seize those opportunities. So undoubtedly the advances in forensic DNA, they've been massive over the years, but along the way we've had some challenges. Initially those challenges were talking about the numbers. They said, well, actually, how can you say that this match was, the chance of this match being 1 in a billion? How can you say that is the case? So that those very first iterations were challenging the strength of the DNA match. Nowadays, the challenge is actually how did the DNA get there in the first place? Could it have been deposited accidentally? So the words we used are accidental or incidental. Yeah. So is it an accidental contamination, or is it incidental to the actual crime itself? And all of that's been brought together by these advances in sensitivity. So I went back on there. I'm sorry. Let me just try to explain it to you this way, and I appreciate, guys, that alphabet spaghetti, alphabet soup, won't be very popular in India. I do love your food, but I've actually never seen this type of dish. It's a dish that we give often to kids. So as children, we're often given this. It's pasta, a pasta that's punched into letters, and then is dropped into, is mixed up with like a sauce. And it's called alphabet spaghetti. Has anyone ever heard of that? Why would you in India? But alphabet spaghetti is probably not a good. I'll have to try to find a better, more Indian-centric example. But can you see that with the problem with a mixture, DNA mixture, is that we know that a particular person actually might be in this mixture, but we can't say sometimes. Because, as you can see here, we can actually make this name out of this bowl of soup. So if that was a DNA mixture sample, we could say, oh, that's John O. suspect. Is that he's a possible contributor to that mixture. But of course, you can actually see that we can make up some different names as well. So that very, very simply is one of the problems now we face with mixtures. We actually don't know what the combination is. I don't know if that's helped or hindered, but perhaps if you could put either a thumbs up or a thumbs down if you probably don't know much about alphabet spaghetti. But it's a very simple way of actually saying what the problems are now with mixture interpretation. And the problems are going to be alleviated by, to some extent, by increasing sensitivity with AI. So let's just have a look now at the latest techniques going toward the end. Speed, we've mentioned already, rapid hit. Imagine being able to do a profile now in 90 minutes. Can you imagine what that would look like at the borders, doing that at the border desk or something like that? Sensitivity we've spoken about. Previous test, that SGM plus multiplex was 1 nanogram. The next generation came around 500 picograms, so it's half the sensitivity. And for around about 80 cells, you can actually now generate a DNA profile. So that's on the one hand, that's good. On the other hand, as you can see there, it gives us lots more opportunities for contamination. So we found this as we'd evolved in our labs, that our labs, although we thought that they were very clean for the day, they weren't clean by modern standards. So what do we do? We know, of course, that conventional DNA testing now, we would say it works very well, it's well established. Sometimes, as you saw with the Amelia case, It's not the science itself that lets the side down, but the way in which it's applied. It's easy to upload and to search the National DNA Database. And I'm hoping, guys, and I think in your lifetimes, you will actually see in India the evolution of a DNA database. I think that's going to come, and believe me, guys, that will revolutionise your crime fighting in the country. But it will come with some problems because lots of people won't understand it. That really is why we need all these your young bright minds to help us make sense of it. But what do we do if we if you have a poor profile, if you don't have enough to match on the database, if we have a degraded sample? What can we possibly do? One of the things we can do is we can move to massively parallel sequencing, next generation sequencing, and it takes the good bits of all of the evolutionary techniques that we've spoken about, and it will do lots of DNA sequences at once. It will start with small amounts of material, and it's for that reason that this is the test of the choice, really, for the bones from Ukraine at the moment. So little samples of bone being taken from these fallen soldiers, sent to the Netherlands, and these techniques being done on that bone. So the conventional DNA looks something like these repeating base pairs, these repeating sequences. You can actually see that we've got a 9, a 9, and a 10. So these repeating sequences will vary between varying lengths, and it's those repeating sequences that we can then use to actually build up the DNA profile. You all know that they combine of the Char-Gaffs, guanine, adenine, cytosine, and thymine, and that's how we actually do it with the sort of standard testing. With massively parallel sequencing, what we're actually doing now, we're focusing down more and more onto to one base pair. I'm hoping you can actually read from the slide. So we're not looking at only the length of the DNA, but we're actually looking at the specific DNA sequence. We're actually within. We call it microvariants. So the more variation we can actually get in forensic science, generally speaking, the more the better the evidence is. So you can actually see there that we get looking at that 5 repeating base pairs. We can actually get all of that information using the same assay. That's the key word there. That same test to actually do it all at once. This is a primer, so I said keep your phones handy. I'll just leave this for a moment. But this is a primer to actually tell you what massively parallel sequencing is, to take you through that through those techniques. Has everyone got the access to the primer? Just put me a thumbs up when you've got the access. So okay, two people been able to access it. I'll just leave it up for just one moment more. So we've got a few more people able to access that. So can we check the condom wrapper also? The condom wrapper is, going back to my first story, the condom, what we were hoping to find there, we were actually hoping to find, of course, the male component of the offender in that condom, the semen, and we were all hoping to get epithelial cells from the outside of the condom. Remember, we didn't have enough semen, we didn't have enough epithelial cells, but you could also, of course, look at, I would probably look at the fingerprint wrapper, sorry, the condom wrapper for fingerprints. We wouldn't necessarily be looking at that for DNA, but good question nonetheless. Okay, guys, so this is the type of kit that we actually need now, and there are some hyperlinks to this equipment. This is the MySec platform. This is the piece of equipment I showed you on a previous slide that is used for next generation sequencing. And again, you see that you can actually access that through. It comes with its own preparation kits. What I mean by preparation kits, I mean a combination of the multiplex, the kits. Those of you who've tried to make up your own DNA extraction kits and so forth will realise that it is massively difficult. Get mixtures and so forth. These kits come pre-prepared, and they're exclusively for the use of next-gen sequencing. We then have the sequencer, MySeq sequencer, and then of course it's analysed in a different way. So that's the piece of equipment that we actually have used to do that. And you'll see that in one test it will do all of these. It will do autosomal STRs, so that the sort of standard STR test that we need for the DNA database. It will look at YSTRs, so STRs that just appear on the male side of the chromosome. The XSTRs, the familial type cases we've spoken about. It will look at some identity SNPs, so single nucleotide polymorphism SNPs. It'll look at biological ancestry SNPs and some phenotype SNPs. And it will do all of that in one technique. So that's the benefit really of that test. We start with extraction the same before, and we just effectively we use different types of kit nowadays. So it was launched in 2015. So it's not next generation sequencing. It's been around some time, but it is very, very powerful nowadays. Can you see that it makes up to 230 markers? So at the minute where we in the UK are looking at DNA 17, this, can you just see how much more discriminating it is, how much more sensitive it is? It's accredited to 17025 standards. I'm hoping that terminology is known to colleagues in India, because all of this must come with a massive health warning that we must apply rigorous quality standards to this. So ISO 17025 is that standard. So when we use SNP sequencing, effectively we're using next-gen sequencing. It's not new, but its application in forensic science is new. We have three options. We use which option we actually use is depending upon the quality and quantity of the biological material. So if it's a poor quality sample, bone from Ukraine, if it's we don't really have a small amount of it, would depend upon which one of these three options we use. So the SNP microarray is not good for DNA, but it's the least expensive. The targeted kit is the mid-range, so that looks at between around 10,230 SNPs. We can start with 50 picograms, so a much less amount of starting material. The cost is mid-range. Can you see with SNP microarray we need 200 nanograms, so we need much more starting material to start with. The targeted kits, we can actually go down to 50 picograms, and we can actually go down the same with whole genome sequencing, so the most expensive, as you can see there. And that's the technique now that's recommended for the most degraded DNA samples. And it does all of these at the same time, as I mentioned before. These are all the benefits. It eliminates the need for multiple STR kits, so we don't have to do this repeatedly. We don't have to test the sample once and then twice with another multiplex. We can interrogate 200 genetic markers. It gives us a range of information from single nucleotide polymorphisms. It's rather put mitochondrial DNA to bed, really, because the same type of result you can actually get from this as you can from mitochondrial DNA. So we can those are the new kids on the block. We've got greater genetic allelic detail. And remember I was telling you before that when we were looking at this site 7, we looked at site number 5 before, looking now at site number 7, GATA. Can you see within GATA we're looking now at these microvariants? We're looking now at much more variation actually within the sample. Where you have variation, generally speaking, it gives you much more benefit. Will the presentation be shared? I don't have a problem with you having the presentation if you want. If you want to email me, that's fine. If you want to just have it, then that's fine also. Do you remember back in 2002 when I was telling you that we were experimenting with familial searching, where we don't have a match on the database, but we have a relatives match, a relative DNA? That's really where we were at the time. Let's just now move on to test your observations. How is DNA profiling used using conventional DNA analysis methods using methods such as DNA 17? Which is it? Is it A? Is it B? Is it C? Or is it D? It's B. Why is massively parallel sequencing particularly effective in cases involving degraded samples? It can only analyse fresh DNA samples. It's less accurate when compared with compromised samples. It can yield accurate DNA from small or aged samples. I think we all agree that that's number C. When comparing a suspect's historical reference sample pre-2004 with the modern DNA 17, how are they compared? Let's go for C again. We can only compare the loci in common. We can't compare an apple with a banana. That's really what I'm trying to say there. The latest from the USA is really taking familial DNA searching one stage further. Remember I told you that the way in which we looked at it was to actually look for family members who were on the DNA database. This is looking at people who are not on the database, who have been nowhere near a database. I don't know if these kits are commonplace in India, but in the UK you can actually buy a kit. Sometimes they're bought at Christmas, and you can buy them in shops, and you can take a DNA sample, a DNA profile of yourself, just a buccal scrape, post it off to a company who will then analyse the sample with SNP technology. SNP technology gives you lots more information than STR does, and it will tell you, if you're a third Indian, if you're half, it will tell you things about your genetic background. And this is the very, very first case, so really takes familial searching to a completely different level. With the case of Joseph James D'Angelo. It was called the Golden State Killer. If you want to scan that QR code, there's a little primer. You can actually see the little document beneath that. Well done, Tisha. You can actually see that it opens up into a little primer, and we'll actually tell you more about how this technique is done that I've got a chance to do in this short time I have with you today. Yeah? Well done, Samantha. I knew you guys were going to be good at this. So this is actually what happened. Some of you actually might have heard of this case, but in 2018, a guy called the Golden State Killer, his name was Joseph James D'Angelo, who actually had been a policeman. In his former years, he was actually a policeman while he was committing these offences. I'll answer all the questions at the end if I can, please. He was not captured for 43 years. He was committing some dreadful crimes. He was breaking into houses, raping women, killing their husbands, and he was nowhere to be found, despite the fact that America have their DNA database. It's called CODIS. It's their database, and there are no matches. What could we do? This person is not on CODIS. In the same way that Joseph Kaplan was not on CODIS, so was neither was Joseph D'Angelo, or had he ever been anywhere near a database. So they went to a really smart, actually, because this is the testing chip that's used for SNP testing. You can actually see that it gives you much more ability to look at variation. Can you see that you can actually look at the numbers of shared centimorgans? That's the unit of currency with this type of test, not repeating sequences, but centimorgans. But you can actually see that you've got lots more opportunity to discriminate, and you can actually look at the location of these also. So they tried to use conventional investigative methods. They've tried to look at STR profiling, no match on the database. What could they do? So they actually went to the overseas authorities. They actually went to they looked at the overarching database. It was called GEDmatch, and they looked at all of these samples that have been submitted to these ancestry companies. So they also used common investigative standards also. So once again they used reverse genealogy. Reverse paternity, we called it back in the day when we were looking at Kaplan. We called it reverse paternity. We weren't looking at we weren't looking for the son of the individual. We were doing it the opposite way around. We were looking for the father of the individual. So I'll just take you through this slide. If you look at Kaplan, so if you look at D'Angelo and all of those dreadful cases, and if you access that prize, it will tell you. It will give you a little podcast. In the primer, you can actually listen to the podcast. It'll tell you all about the case. It's run initially through the CODIS database. It doesn't give us any match. Then the GEDmatch databank is used, because GEDmatch is that database which is used by lots of these ancestry companies, and it looked for some shared DNA and cousinship, guys, really is the key to this, because you'll all remember, you'll all perhaps reflect that if we get 50% of our DNA from mum and 50 from dad, then we must get 25% from grandparents, about an eighth from uncles and so forth. So there is a diminishing similarity all the way up either side of your family tree. My slides have gone unfortunately again. Let me just reload. Sorry guys, my slides just disappeared then. We're almost coming toward the end now. So that's where we were, and we went up either side of the family tree. Can you see that point of common ancestry, the paternal grandmother? Yeah. Can you see from the other side that the cousins? So we start with those two cousins, and we go up to the point of common ancestry. So we've got this SNP profile. We then have this family tree. We then have to turn the SNP profile back into STR. Actually, a sample Joseph D'Angelo was never near a database. So the cops followed this guy down at the street one day, and they picked up a drinks can. He'd thrown a drinks can down. They swabbed the drinks can, and they matched him then to all these cases all those years ago. Now I hope you'll all agree that that's smart. Let's just have a few thumbs up for the Americans, because that's even taking it one stage further than we took it. That really is smart. So I'm going to now just finish off with one last case, and this last case ends really with it almost should have a subtitle, because I would have thought, who would have thought this when I started in forensic science? Who would have thought that you could have ever got any form of usable forensic material from a brick? and anything such a thing as a DNA database that would actually match this person to a suspect. Let me just tell you a little bit about this story, because this was the first case where we used low copy number DNA, so super sensitive DNA of the time. We'd actually used a database, and we'd actually used familial DNA searching all in one. And this is a story where two brothers went out drinking at night. They got drunk. They tried to break off a car aerial, and they dropped a little bit of blood on the car aerial. They then walked down the street, and they picked up some bricks. They were like bricks that you have for building a driveway, a block driveway. They went to the bridge overlooking the motorway, and they threw them over the bridge, threw one of these bricks over the bridge. It smashed against the window. It actually hit the upright, spun the brick into the chest of the poor man, who was eventually killed. He was killed. This is some of the photographs there. They're not graphic, but that's the bridge where the boys had thrown off the brick. Can you see just down in the distance? Can you see the lorry where he'd actually managed to bring the lorry to a standstill, put the hazard warning lights on, and then he'd actually died. He'd passed away in the cab. You can actually see there where the brick hit the upright of the lorry, hitting him in the chest and killing him. And are you with me, guys, in thinking there's no chance at all that we can actually get some biological material from that? So they did get biological material using the low template DNA. So they got a DNA profile from the brick. Who would have thought that back in 1986 when I started? Then what do? We have no one on the database who matches. Can we do a familial search? And we did the search. So this is the sibling search. Remember I said there was a parent and child search, the one that we'd actually done in the Kappan era. We're looking for the Kappan through his son. We can also look for similarities amongst brothers and sisters. So do you see, at the time we had 16 markers. So can you see the alleles? To the top one is 16 out of 20. So what that means is that 16, this person does not match on the database. He can't say that he matches against the sample. But there is someone on the database who actually matches 16 out of 20 of those. And the higher up the list you get, can you see we've got 15 out of 20, but the locations were different. It's a bit like finding these matches, but one is in Delhi, one is in Ahmedabad. You actually put the Delhi ones more closely together. So we had two profiles, 16 and 20, and one of them was the offender, Craig Harmon. We looked at the closest siblings to the scene, and he was actually arrested. That is effectively a summary of the case. He was the only person who shared 16 out of 20. Remember, he wasn't on the database, but when he was arrested, of course, you take a buccal scrape, and then you make up the full sample. He was arrested just over the border, so he wouldn't ever been sampled. He wouldn't have been sampled, but he had a brother who'd been arrested before, and that's how he was captured. So, thanks for asking me to speak with you today. Let me just check what this opens up into. This just opens up into my bio. I thought it opened up into something more useful, but that's my bio. If anyone wants to get in contact with me, and I think this last one, this last QR code, if you just scan it. This opens up into a little, I suppose it is almost a feedback form. You can rate the presentation, you can ask questions. So if you actually want to use that last slide, the QR code on that last slide, to ask your questions, that's where you'll find it. You see, we've got some hands up. I can't see what questions are being asked though. Let me see if I can look at the chat. Let me just look at the chat, please. I'll go back from the bottom. Identical twins do have the same DNA, yes they do. But if you want to look at we have a Facebook page for my university. It's called SPS Forensic. So if you go onto Facebook and you actually look at for SPS Forensic, if you like and follow that page, you'll only see from, I think it was last week or the week before, where the French had actually managed to identify two brothers, two completely twin brothers, using DNA. My LinkedIn, I am on LinkedIn, so it'll be just something like Robert Green. I'm not under no pseudonym or anything like this. Genealogy is a combination of genetics. So lots of what we've talked about already is genetics. Genealogy is the type of thing that looks at family trees. So the genealogists will actually, I'm pretty sure that some of you may have even done some genealogy yourselves. You're looking at who your grandfather was and his grandfather and so forth. So that's what we mean by genealogy. And these new techniques link, of course, genetics and genealogy together. So hopefully that was. Should India mandate judicial warrants and independent oversight committees before conducting familial searching? It will need some ethical oversight for sure, and we had lots of scrutiny when we were doing familial searching. I mean, some of the cases I've shown you are of really good. They were good quality matches, but realise also that they come with some form of intrusion, don't they? It's an intrusive. Can I highlight career opportunities available in genomic analysis? Let me just look, please. I don't know off the top of my head, but if you put your question in my in that little chat box that I've just given you, you can actually see it on this slide. I'll answer that. So identical twins have the same, well, genetically identical twins do, but not the heterozygous twins don't. I can see the webinar feedback form, which Pushkar has put up. How do we get the PDF? I mean, I'm happy with you having it. You can, if you want to put your details on the in the chat. I'll do that. Have I missed anything? So would India mandate some oversight? Yes, I think they would, for sure, because you need some rules around this. And of course, with the evolution of DNA, you've got. I know that you've actually got 3 new laws in India, where you're looking at developing more and more use of forensic science. That's great, but of course it also needs more forensic science means more understanding. So we actually need you guys to help people understand this, because otherwise the science will work and everything else will fall apart. So that's really all I've got for you today, guys. I hope that some of that was of interest. If anyone wants to ask a verbal question, I'm happy to try to do that. Thank you, Nikita. Link send again, please. Link send again, please. Certificate. I don't know what you mean by that. You're very welcome. link send again, please. Thank you. I'm hoping to visit India. I generally come in summertime, so once my teaching ends, I'm generally over. Yeah, look out for me. Generally around about the summertime. I've been actually once already this year. Probably come about 3 times per year to India, and always love my time there. Always love your food and your culture. So thanks for inviting me. Shall I now turn off the stop sharing? I'll stop presenting. If you want to put on your cameras, and I can see who I've been talking to, it doesn't matter if you don't. None of my students ever put their cameras on.
Thank you so much, sir, for giving your valuable time to our team, for our participants.
You're welcome. Okay, so thank you. Thanks once again, and I look forward to seeing you soon.